Good evening, ladies and gentlemen, colleagues, I see, friends. I'd like to welcome you to the IPFW College of Arts and Sciences Distinguished Lecturer Series. I'm Dr. Mark Littman, the Dean of the College, and it is my great pleasure to welcome you. As does any great university, IPFW provides a wealth of educational and cultural benefits to the community, in addition to the classes we teach and the research we do. That's part of the mission of the university. That's part of the mission of the college. This lecture series, which is sponsored by the college, is a rather potent example. The series began in 1982, highlights the breadth of disciplines that comprise the college, comprise the arts and sciences. And as you can see by looking at the titles of previous lectures printed in your program, we cover an enormous range of topics. We do this twice a year. In the fall, we bring in an outstanding external scholar and speaker. In the spring, tonight, we celebrate an internationally renowned scholar from amongst our own faculty. This evening, we have Dr. Bruce Kingsbury. He is professor of biology. He's the director of the Center for Reptile and Amphibian Conservation and Management, usually called the Herp Center. What that really means is he hangs out with snakes. And he is the chair as well of the IPFW Department of Biology. He has degrees from Pomona College, San Diego State University, as well as University of California. Dr. Kingsbury has dedicated much of his professional life to the conservation of endangered reptiles, especially snakes. His areas of expertise include habitat requirements, population viability, habitat restoration and management. His areas of interest also include behavioral and physiological ecology of reptiles, their thermal regulation, foraging behavior, and patterns of habitat use. He's the author of numerous articles, books, videos about snakes and amphibians in Indiana in particular. He's the recipient of numerous grants to work on, among other things, the ecology of the Massasauga, which is an eastern rattlesnake. And in fact, I'm pretty sure he told me that every person in attendance would receive a personally autographed Massasauga. He's used a lot of that grant money not just to carry out the work of the center, but to support graduate students that carry out the work of the center. So there's some synergy between the educational efforts, the research efforts, the contributions to our knowledge of ecology, and so on, all as a whole. That's part of what makes a center of excellence a center of excellence. In fact, the whole thrust, I believe, is, if you look at the title line down there, Bruce Kingsbury and the K Team. So in his mind, it's very much a case of what the synergy of the center does as well. In any event, as you undoubtedly already know, he's going to be talking about snake tails, chasing reptiles, and promoting their conservation. Dr. Bruce Kingsbury.
Well, yes, that would do as a sound check. I can hear very well that the microphone is on. Well, thank you very much. Thank you for coming. Thank you very much to Dean Littman for a very nice introduction. Some people have asked me if I was going to bring any snakes. So So I didn't bring any creatures, but I hope you'll find it entertaining anyway. I do want to use this talk to say thanks a lot of times, and it's very rewarding to be here, and very nice to see you all here, and I appreciate that, and I also want to say that I appreciate the opportunities I've had here at IPFW to do what I've done. So with that, let me start off by saying just a little bit about snakes and humanity. We certainly do have a fascination with snakes. They've shown up in a wide variety of ways. within our culture, and we have seem to have an innate fear of them. I would love to put a transmitter in one of these, but so far we haven't come up with one. They enter our art in various ways. I have no idea what species this is. And also in our forms of entertainment, I just got online and I started surfing around trying to figure out, well, how do snakes enter into our culture? And this is a site where people are playing games. And can anyone tell me how to get past the snake monster in the temple? So then there's some helpful hints down there as to how to do it. Snakes are feared in around the world, and 99% of the time for no good reason, I would feel. Here's a couple shots of dead ones. The one on the left with the little girl proudly displaying a snake that she had shot this. I think her father had gotten her to do this. I think this is like a rat snake or something like that, but she's been trained from a young age. The other one is a commonly used, it's a real dead snake, but I've seen this same dead snake attached to several different people in various poses. If this was a real correct size, this snake would probably, it might weigh 30 pounds, 25, 30 pounds. And as you can see, the guy's having no trouble holding this thing up. It's a constructed photo. This same snake was also killed in Michigan. This one, I think, was killed in Louisiana or something like that. So this snake shows up now and then. I don't know who that guy is. So why am I interested in snakes? And I thought about that a little bit, and I'm really not sure, but it's for sure that I've been interested since I was about 5 or 6 years old. And my parents were pretty accepting. My dad did try to convince me to enter a different occupation, but not very hard. And I do have a couple snake tales I wanted to tell you about from when I was a kid. I'll keep experimenting with where to stand so I get rid of that reverberation. But the first tale is about the insurance salesman. I had a really big king snake for a pet, and I was sitting on a overstuffed chair, and the snake got in and started crawling into the chair. And so I didn't want to hurt the snake, so I let it go, and it crawled into the chair. And I figured, well, eventually it'll come out, and I'll pick it up again. And it did, but it came out while an insurance salesman was talking to my mother in the living room, and it just started crawling across the floor while they were having a conversation about some plan. And my mother, who was used to this kind of thing, said, "Oh, there it is." And she got up and she picked it up and she put it back in its cage. And very soon after that, the insurance salesman left. But and then there's the gourmet meal story. This one was a Thanksgiving where everyone was in the living room enjoying things, getting ready for sitting down to a nice meal. And then my dad called us in, and he had taken frozen rats from the freezer that we had thawed to feed the boa constrictor that you saw in the other picture, and he had put one on each person's plate and then invited them to dinner. So only in a house where you have a budding herpetologist. Right, well, I do think snakes are really quite fascinating. They're extremely variable. They have a wide variety of shapes, colors. Some of them are in fact very dangerous, and some are not. They do cool things. It's pretty neat that you can get about the world without any legs, and they do it in a variety of ways. They are just derived from lizards, so they're essentially lizards without legs. But they have figured out how to get about without the use of appendages. They have a lot of interesting kinetics in their skull. They have a large number of junctions between different bones have very flexible articulations. And so we have essentially a single system to open and close the jaw. Well, snakes can do all kinds of disarticulations and flexes, and one of the outcomes of that is they can have fangs, which when they open their mouth, it opens the fangs at the same time, so they don't bite themselves. Everything folds up nicely. This just shows a variety of forms that have that, but as they open their jaw, that pushes forward on a bone, which actually flips the mechanism out and allows them to bite whoever they're chomping on at the moment. It does other things as well. It allows them to eat extremely large food items. So here's an image of an egg-eating snake, just about done downing one. So you can imagine that this when the snake is just at rest, the head is really only about that big. And so this guy's got his head and mouth around a large meal. Now they actually have modified vertebrae that pierce the egg and they crush it. They don't just squeeze it because, of course, if you've ever tried that, you discover that eggs are pretty resistant. But if you puncture holes in it first, then you can crush it quite easily. We do have some venomous species. Almost all of the snakes anywhere near here are non-venomous and not dangerous at all. They might hurt you if you pick them up because they think you're trying to eat them. But we have the Massasauga, which I'll spend quite a bit of time on this evening. There's a cottonmouth known from maybe just one location in southern Indiana. All the other water moccasins that people see are just the northern water snake. The timber rattlesnake in some areas in southern Indiana. And then our most common venomous snake is the northern copperhead, which is in the southern half of the state. All of these species are pit vipers, and here's, if you can see my little pointer up there now, here's the nostril. Back behind that, on either side, is a heat-sensitive pit, so they can actually visualize a heat-emitting object, much like they could with eyes, like we can. And then, of course, there's the eye behind that, and the snakes in this group all have vertical pupils, and so if you get close enough, you could actually, and you were in Indiana, and you weren't looking at somebody's escaped pet, and it had eyes like that, you would want to be cautious. But of course, once you go somewhere else, that rule doesn't apply. But all of ours are pit vipers. And here's my token tail in my tails for today. This is a Massasauga tail, and this is the rattle on the end of the tail. These snakes, at least until recently, even occurred within the city limits of Fort Wayne, but they may not any longer. But it's a very diminutive snake, but sure enough, they have a rattle on the end of the tail. They do have a business end. I would never handle a snake like this unless it was out cold. This is a snake that's been anesthetized for implantation of a transmitter, and so it's very cooperative. But if you open the mouth, you can actually pop out the fang. So there's the business end. So there's a little bit about snakes, snake stuff, and now I want to turn to some stories about the work I've done along with my students. I want to say a few more thank yous, and then I want to go into a little bit about the types of techniques that we use, and then I've just picked a few things to talk about in a little more detail, and we'll just kind of pick those off one by one. Before I get in that, though, I want to say a couple more kinds of thank yous. I have been very well funded since I've been here. I've received support from IPFW. I've also received quite a bit of support from a variety of state and federal agencies, and also NGOs, non-governmental organizations like the Nature Conservancy. And it takes a fair bit of cash to do the kinds of things that we're talking about, and I've been very fortunate to get very good support. And the other thank you, I want to a shout out to the grad students. And this is just a sample of the K team. Almost all of the field work these days that gets done is by the grad students and undergraduate assistants. And so they are definite collaborators on everything, and I want to make sure that especially the guys who are around today and those who have been through here before know that I really appreciate the work that they do. So GIS, Geographic Information Systems, allows you to visualize spatial environments. You can do anything that has a spatial component, you can use GIS on. So you could look at populations, you could look at habitat, you could look at interactions between a variety of different sorts of spatial data sets. This is just an example of work done by a grad student, Michelle Standora, who modeled a variety of components in the habitat in the present and in the past to look at where Massasaugas would be across the lower peninsula of Michigan. This just shows you the southern half of that. It also allows you to visualize habitats that you're doing other work in, so you can look at proportionalities in terms of what the animal likes to be in versus what's available, that sort of thing. And this is supposed to be a little bit about reptiles besides snakes, so I've stuck in maybe 2 slides of other reptiles, but it's pretty snake heavy today. But here's an eastern box turtle. Basically what I wanted to convey to you here is if you're trying to figure out what this turtle is going to do, we can't talk to it, and we can't even go out and look and find it very easily. If you walk away for an hour and come back, it's probably not where it was, and you'll never see it again, maybe, or at least not very often. So what we do is radio telemetry. For snakes, we actually stick the transmitter inside the snake. For turtles, we can stick it on the outside. And so there's a transmitter, and there's the antenna trailing off the back. And once you do that, you can go find the animal whenever you want. And then I send someone else out there into the mosquitoes and ticks, and et cetera. And they track these guys around, and they come up with observations. These are just observations that are collected over time. And then you can analyze these by a variety of methods and come to your conclusions. It's a very time-intensive approach, but I really love it because it really tells you a lot about what the animal's doing. That if you just went out and looked for them, when you would find them might not have anything to do with what they're actually doing in terms of the reality of the situation. It also lets you see the animals in their natural environment over and over again and visualize things that normally you wouldn't get to. And in this case, here's a Massasauga with these red things on it. and it's pretty hard to see from there, although there's a clue in the picture. But if we go a little closer, those are mosquitoes. So Massasaugas have a problem with mosquitoes too, just like we do. I think this one was holding extra still because we were only a few feet away. But you get to see all kinds of really neat things. Here's a pair of Massasaugas where the male is courting and guarding the female. He's much larger and he's wrapped around the female. She's sitting there in the middle. And next year, after they get together, we have this sort of situation. Here's the mom in a burrow, and here's the young that she's given birth to. So if you don't have telemetry, good luck finding most of those sorts of observations. Okay, so now time for some real stories. First thing I want to tell you about is some work on copperbelly water snakes. This is Nerodia erythrogaster neglecta. This is a federally threatened species, so it's federally listed. And the range for the species is down here in this tri-state area to the south, a little bit around Seymour, Indiana, and then up here, closer to home. And these are actually the listed population. So if we go in, zoom in on them, this is their range perhaps 50 to 70 years ago. This is the range now. And in fact, that's a bit of an exaggeration. If you go to the sites where we have been able to reliably find them, just like 6 places, the number of snakes that we've been able to find is just going down and down and down. So the species is in trouble, and that's why it's listed, and that's why we're funded to do the work that we do on them. And one of the things that we've done is we've done comparative work between the copperbelly water snake and the northern water snake. And this is a project that John Rowe in the audience was involved with a number of years ago. John's back with us as a postdoc, but some of the stuff I'll be showing you now is from the work that he did. And the copperbelly is in trouble, as we showed, and then the northern water snake is ubiquitous. You find it all over the place. You find it right out here in the river, out in front of the school. Found it downtown, Fort Wayne. So these are 2 species. They're closely related. They do very similar things in a number of ways, and yet one is on the decline and the other is just fine. So by comparing these 2 in a place where they both occur, we can learn something perhaps about why one's in trouble. So we did that in northwestern Ohio, and here's a sample of the kind of data that a lot of the students generate in a variety of studies. This shows these polygons here are for northern water snakes. That's the common one. And then these polygons out here are for the copperbelly. And what they're doing is they're minimum convex polygons. They're just encapsulating the outermost observations to let us talk about how much space they use. But you can start to see some things right away. One, the area occupied over the course of a season, which is what's depicted here, is much larger for the copperbellies compared to the northerns. You can see that the northerns don't travel to as many wetlands, and you can also see that the number of observations in the uplands is far greater for the copperbellies than the northerns. Well, if we go to another place a few miles away and do the same thing, we see the same pattern. These snakes, this individual snake is going to this wetland, this one, it's spending time up here, it's over in these wetlands, different places over here. This northern is just staying in that wetland, more or less. So that leads to a variety of observations, and this is just them boiled down. And I have these grayed out because I'll come back to them, but the first set of observations is the number of wetlands used by copperbellies is basically twice as many as the northerns. The number of times they move back and forth between the different wetlands is 3 times, which means they could go to one and then go back to the first, so they're just shifting more. The shift distance, that is the distance they travel from one wetland to another, is on average about the same, but the biggest moves are made by copperbellies and more small moves are made by northerns. There's a much more profound use of small wetlands, and if we look at just ephemeral wetlands, that is those wetlands that dry down during the course of the study, they were ephemeral or temporary, it's also relatively different. Now, if you sample wetlands in a natural system or even in a system that's been a little bit modified, what you'll find is that most of the wetlands are actually quite small, and they're pervasive. The problem is they're also really easy to drain, so they're the first to go. And so when you look at the topography now, at the landscape now, you have more large water bodies, and it looks like that's the way it always was, but in fact there's tiles underneath the ground, there's ditches, there's all sorts of things in order to get rid of those. Well, if you start taking those out, you start increasing the distance between the wetlands that are next to one another. So not only are you removing a certain type of habitat, you're also changing the interplay of adjacency and the collectiveness and the collective effect of these clusters of wetlands. And over here is a graph which shows the copperbellies and the northerns compared when you look at the average distance between the wetlands, and then I guess we're clipped off a little bit here, but essentially this is a log of the size of the minimum convex polygon. And what should be quite evident is that if you look at the northerns, there's no correlation at all. But if you look at the copperbellies, there's actually a pretty nice curvilinear relationship, which tells you that as you have wetlands farther and farther apart, you have the snakes are going farther and farther to occupy that area up to a point, and then that plateaus off. So you're going to influence what they do, and you're going to impact how they do. If we look at the bottom half of this table, upland habitat use, that is not wetlands, but uplands. The copperbellies use an order of magnitude more upland habitat than the northerns do, and they tend to be further away from the wetlands. And then if we look at a measure of total area used, like the MCP, they're using much larger areas. And if we look at some measures of the movement distances that they make, not the distances between the wetlands, but the total distances moved, they're moving twice as much. Well, that means if you start putting all those things together, that the landscape that the copperbelly is trying to use has to be interconnected. It has to have these wetlands available. It has to have safe corridors. It has to, the less habitat that's available, the further it has to go. And those kinds of landscapes aren't available generally. So now we start to understand why the copperbelly is in decline. The other thing is, the farther you have to go in northern Indiana, the more likely you are to run into a road. I mean, everything's pretty much gridded out here, even out in the country. And in fact, for all the modeling we've tried and everything, if you just look at a map and you ask where's the forested area with some wetlands with fewer roads, well, that's where the snakes are. But this helps you understand why. If road density increases and you have a snake which moves further, then this is a modeling exercise. What it shows you is that the copperbellies are going to have to cross the roads more and more frequently. Well, that's true no matter what the animal does, but for an animal that doesn't move around as much, like the northern, that's going to have a much smaller significant slope, and the outcome is that if you do, and this is work that John did, you can see that if you have a certain number of cars, a certain number of tires, certain number of lanes, or however you want to do it, you can get your calculation there, and then you can figure out, well, what's the chance of a snake going to have crossing the road? And what becomes apparent is quickly it becomes a significant factor, so that you can have a dramatic effect in terms of mortality. So copper bellies are in trouble for probably a wide variety of reasons, including the loss of these ephemeral wetlands, their dependence on wetland complexes as opposed to isolated wetlands, and also encounters with things like roads. And if we want to save them, we're going to have to respond to these threats or these needs. All right, so there's story number one. Story number two is Massasauga hibernation ecology. The eastern Massasauga is one of those venomous snakes. If you're going to have a venomous snake in your area, this is the one to have. They live in areas where nobody wants to go. They have been shown to rarely bite even if they get stepped on, so they're very shy, not aggressive whatsoever. And actually, because of their small size and the fact that their venom isn't really super toxic, if you get bitten, you don't want to get bitten. But there are no records of mortalities, maybe period, in around here. But in the last 50 years, there haven't been any mortalities in Indiana, Michigan. I don't think in Ohio or Illinois either. But still, would be bad to be envenomated by Massasauga. You would not be happy. But I digress. Getting back to hibernation, their hibernation sites are affiliated with wetlands, and the thinking there is requiring access to groundwater, and I'll get into that a little bit more. But here's a Massasauga just coming out of the ground in the spring. And this highlights work done by Chad Smith, who's here this evening, and also Joe Sage, who graduated several years ago and works as a biologist down in Florida. Most populations that have been studied occupy areas that have crayfish burrows, and that's where they hibernate. Here's a crayfish burrow that's actually active. It has a crayfish living in there, and the crayfish digs down in order to get to the water table and flips the mud out. And so that's why you have sometimes even a chimney of mud that the guy has stuck out there. And the Massasaugas use the burrows that have been abandoned. If you actually go into an area that has been occupied by crayfish, there's active burrows and inactive burrows. And if you think about it, if you stick a hole in mud that's of a firm type, that hole will stay there for a long time. And here's a snake that hibernated in here. This area was burned recently, which explains the sort of the charred look in the background. The snake, though, came out after the fire. It looks kind of white because it has calcareous, a calcium kind of a coating because of the type of soil that it was hibernating in. The other thing about these sites is that they appear to be somewhat limited. The right combination of characteristics appear to not be occurring everywhere. And one way of depicting that is a site from a site north of here, still in Indiana. This is a two-lane highway. Here's a semi to kind of give you some perspective. There's a house. and here's just a country road, paved country road. And these are the MCPs for different snakes within a particular season. And what you should be able to tell with these is that they all cluster back. They have an apex back in here, which is about the size of the stage or smaller, and that's where they all hibernate. So they're not hibernating all over the place. They're actually quite selective about where they hibernate. And you can imagine that if this home was constructed right there, it would do a job on that population of snakes. It probably would also lead to an upset homeowner, at least for a season or two. So we were looking into that, and we've begun to study that relatively intensively. And we wanted to characterize these hibernacula and figure out why they like certain places and not others, and also look at what the snakes are doing, and then hopefully we could provide guidance to help protect the snakes by protecting this vital resource. So the methodology for this includes water chemistry studies, which I'm not going to talk about in soils and all kinds of other goodies. But the most straightforward thing is to use radiotelemetry to find where the snakes hibernate, and then stick a fiberscope down in there and see what they're doing. So Joe Sage did quite a bit of that, and Chad has been at it. I can publicly apologize to Chad as the only student of the 25 that I've been involved with supervising that I have never gone with him to collect data during his field season. And maybe you'll at least understand why when you look at this. Hibernation occurs in the winter, and Chad's study site is in the northern Lower Peninsula. I have to admit that he probably does have fun. I don't have any snowshoes. But he did have to go out there. Some of the data collection is automated, but some of it actually requires somebody going out there and looking down in the holes to try to figure out what's going on. So some interesting factoids about Massasaugas and hibernation. One thing that I think is really neat, and this turns out to be true of copperbellies and northern water snakes, a variety of snakes, in fact, a variety of other reptiles, amphibians, insects, all kinds of stuff, is they actually hibernate underwater. And this certainly keeps them hydrated, but it also probably helps them avoid freezing. It seems counterintuitive, but actually having water in the soil keeps the soil from, slows the soil from freezing. So it gets very cold, but doesn't freeze. Now they are under there for nearly half the year, and I've always appreciated working on snakes in the Midwest because you don't actually have to do too much of them unless you're Chad in the winter because they're underground. But they do eventually come out, and that's in March or early April. But this is a really nice picture, too, showing a snake that's kind of being forced up in the spring. Here you can see that you have leafing out occurring, but you also have snow. So this snake it actually has a little bit of snow on it, but it's not frozen, and it's actually fine, and it'll come out in due time. They do hibernate underwater. Here's a couple of years with average water table depths where the snakes hibernate and where the snakes are. And probably in the sites where we work, they actually stick their noses up periodically and take a breath, but they sit underwater. We do have work from other sites where the snakes have actually been completely inundated. There's no way that they could come up for a breath of air, but they lived. So they can actually get enough oxygen if the water is really, really cold and really well oxygenated. They do get cold. So here's over the course of the winter, and temperatures start out, well, cold already, but get pretty close to zero And this is actually good for the snake because the snakes can't eat during the winter, but they could still consume energy. And so by being really cold, the fact that their energetics are thermally dependent allows them to utilize minimal amounts of energy while they can't feed. So it's actually useful for them to be as cold as possible without freezing, and they certainly achieve that. They have company. A common cohabitant in burrows are the snakes of the genus Thamnophis. These are garter snakes and ribbon snakes. And here's a great picture of a garter snake coming out, or at least periscoping and checking things out, and there's still snow all the way around the burrow. This kind of snake, if you get close, can just drop right back in. But it's pretty close to emerging. And they also will hibernate with one another. Here's two Massasaugas getting ready to come out. Chad has found 17 garter snakes and 4 Massasaugas in one hole. And it used to be thought that usually there would be one snake, maybe 2. But Chad has some data now showing that they can get quite a few snakes in there, and I think 7 was the limit for the number of Massasaugas in a hole. So that actually is pretty neat if you're into Massasaugas. There's other stuff down there like racers and frogs and a variety of invertebrates as well. Another thing we want to know about is when the snakes come out. Why do they choose to come out when they do? For one thing, it's interesting to just understand why things work the way they do, but the other reason is that land managers often have activities that they want to do, like burn in order to clear brush or that sort of thing. Well, if the snakes are underground, then you can burn right over them. But if they come out, then that's different. So if you can predict when they come out, that's great. So just to give you a little more data, I promise not too much. Here's the legend over here, but basically here's the story. The deepest site down into the ground is the warmest during the winter, and then as you get closer and closer to the surface, here's 10 centimeters down, you have the coldest, and that makes sense because you have the cold air mass over the top. Well, then in the spring, you get a shift. Things start to warm up, and the ground, which has much more thermal inertia, stays colder longer than the surface does because the air is warming it up, and you get an inversion as well as an elevation in the temperatures. And when you see a pulse like that, we're starting to learn about how the snakes come out, and then things cool off and things are kind of quiet, and then it starts to warm up again, and then everyone else comes out, and by here, they're all done. So that's data for Chad's Massasaugas. Here's some data for box turtles from Joanna Gibson's work. And this is the same sort of thing. There's an extra line on here. This is air temperature. And as you can tell, air temperature fluctuates even more, and it can get pretty darn cold. But you see the same sort of pattern, that layering like this, and then it starts to flip in the spring and warm. And then these columns here, and that's the numbers for those columns, show you that the turtles are actually starting to come out. And we know that they come out because we have data loggers on the turtles that collect data, as well as these data loggers in here. So we actually know when they come out of the ground. So that we get this big pulse of turtles, and then cools down and fewer of them bask, and then it warms up again, and then they start basking again. But a lot of them popped out here. The lines here are when they burned these sites. So what this tells us is if they had just burned over on this date, they actually could have avoided those turtles, and some of them were impacted by that burn. Same sort of data for the next year. Again, things are rising up past about 5 Celsius, and you have that flip or that inversion, and everybody comes out. Here, the fire is right before that happened, so that actually had less of an impact. So some nifty hibernation conclusions. The Massasaugas and a variety of other snakes hibernate underwater. They don't freeze, although they get quite cold. They have lots of company, and we are beginning to learn when a variety of these animals start to come out, and it has to do with this inversion and general warming. All right, next story. Entitled Growing Snakes, but this is about head starting and translocation. Head starting is when you take an animal and give it a head start by putting it into a captive situation where you give it as much food as it'll eat, that sort of thing. You keep it nice and warm. It doesn't have to work as hard. And we want to learn about head starting and translocation, which is the practice of picking something up and moving it somewhere else on purpose for copperbelly water snake conservation. But we wanted to do this with a surrogate species, the northern water snake, which is not endangered. So we acquired a bunch of pregnant females. They gave birth to, as you can tell, lots of babies. And we raised them and continue to raise them in essentially drawers where they have all their needs taken care of, at least superficially, and they grow up. And then the question is, what happens if you take these snakes and you translocate them into a site with the intent of doing things like replacing lost populations or augmenting failing populations? Well, what happens? So that's what this kind of study is about. And this is the work of Mike Frank and, again, John as postdoc. And so this is some of their tales here. This is the site where the snakes are being released. They're being released with radio transmitters in them alongside of resident snakes that have transmitters in them, and also with alongside snakes that have been taken from a few miles away. And so you have snakes that know where they are, snakes that grew up in the wild but no longer know where they are, and snakes that grew up in a box and turn out to be relatively clueless about a lot of things. They were all let go in clusters so that we can remove the argument that, well, you let that snake go here and you let that snake go there. Well, we let them go in clusters around the residence. And here's some interesting findings thus far for this work. This has to do with growth. You can measure growth in terms of mass or weight or in terms of length. And so captive reared translocations, so these are the head starts that are put out into the habitat. They're actually being put back right where their mothers came from. So they're not being moved around, but they've never been there. There's the residents, and there's the ones that have been translocated from somewhere else. If you look at the growth in mass, there isn't any growth in mass. In fact, sometimes there's a loss in mass of the head starts, whereas the translocated and residents are adding a gram a day. So then, in terms of growth, in terms of length, the captive reared head starts do better, although certainly not as well as their competitors. But this makes sense because snakes can grow based on stored nutrients. So they're very happy when they're put out, and so they continue to grow in terms of length, but they don't grow in terms of mass because they're not eating anything. So they're staying inactive. They're not foraging very effectively for the most part. And you can also look at who survives and who even stays in the location. The head starts actually survive pretty well, at least until hibernation. But that's understandable if you don't ever come out of a hole and you don't ever do anything. You're going to be pretty safe. The residents do so-so, and the translocated snakes take some hits because, for a variety of reasons, one has to do with mortality. Another has to do with the fact that they actually leave because they're looking for something familiar. So they travel much greater distances than the residents do. So if we're trying to grow snakes and let them go, we want them to do well. So maybe there's something about what we're doing that's not effective. So part of Mike's project is to try enrichment, where you, instead of keeping them in a shoebox, you actually let them go someplace where they can bask on a rock. You can see the fish down here. They can hunt for fish. They can swim around. They can crawl around. It's like a snake party room. So that's pretty good compared to captive unenrichment. Here they have the temperature is set for them. There's a water dish they can sit in. They can pace back and forth in here if they want to, but that's it. And this kind of stuff has bearing on what you're going to do if you're going to actually use these techniques. You don't want to raise a bunch of snakes, put them out, and they all die. But if this technique works, then you'd want to do it. But of course, you can't do it in the same space if you do this technique. So we want to learn about these different things and how they do. Another thing is to try to put them in areas so that they don't have to figure out where to hibernate. We're going to show them. In fact, we're going to tell them where they're going to hibernate. We're going to build hibernacula and stick snakes in there. So here's is this thing called a dingo? Is that what? Yeah, this is the John's driving this thing through the forest, and it's going to dig holes. And here's the dingo in action. So there's John. There's Mike in the beginning of a hole. Here, oh my God, I forgot his name. Phil Beaveridge standing down in the hole and right before we buried him. But then you actually fill it up with cobbles, and then for the most part these were dry. But in the winter the water table comes up. In fact, the water table went right to the top. And then there's a water quality monitoring well. There's a temperature monitoring well. And then you cover the thing up and hide it so that the next troublemaker doesn't try to take it home or just wreck it. But that's a study that's underway now to see, can you even make snakes interested in something artificial? Will they live through that? Will they come back the next year? If you can anchor the head starts in your landscape, you can decide where you're going to put them and take care of some of their issues and succeed perhaps better. So that's the story there. Now all these sorts of things are underway right now, so we don't even know fully what's going on, except we can still say that head starts are not very bright. So a few conclusions. Growing the snakes is the easy part. If you translocate snakes that have some knowledge of where they were before, they're confused. Snakes have a very good understanding of where they are. And the head starts are not so bright, but maybe we can train them into scholars. A last thank you. I really appreciated the opportunity and appreciate the opportunity to have the Herp Center and as one of the centers of excellence here. I do want to say that there's a website which I'll show you at the end for those who want to find out more about it. and there's always nice goodies there about reptiles and amphibians. So there's the website for that, and with that I'll close and give you contact information and be happy to answer any questions you might have. So thank you.
Questions? You really get frogs to live in with the snakes?
Yeah, in the hibernacula, it's pretty neat. It's nice and layered. The Massasaugas are relatively inert, and they're pretty far down the burrow. Then the garter snakes are piled up on top of the Massasauga, and they're a little bit squirmier, so they're the upper layer. And then you might have like some frosting of frogs or something like that on your stack. And for one thing, the snakes are totally incapable of doing anything except crawling really slowly, so the frogs are safe. But the frogs get out of there before the snakes do. So they're lined up in order of who's going to leave first.
As we often do, we like to give the speaker a little something to remind him that he did this. So I have a modest little plaque that says, Indiana University, Purdue University at Fort Wayne, College of Arts and Sciences, in appreciation to distinguished lecturer Bruce Kingsbury with today's date. And I want to give that to you because that's all we're giving you. Thank you very much.